All right, I think we're gonna kick it off now. Good morning, everyone.
Morning.
We are very lucky today to have with us Dr. Juan Antonio Vizcaino. Before I get into the introduction of him, I want to go over quickly some consortium business. So the first thing that I'd like to do is thank all of our sponsors. This couldn't happen without them. We have got the consortium major sponsors: Thermo Fisher Scientific, Bruker, SciEx, and Lilly. And then we also have in the next tier some additional consortium supporters: Agilent, New Objective, Pfizer, MSC, and Impro. I want everyone to know about an exciting upcoming symposium, the third International Top-Down Proteomics Symposium, University of Hamburg in Germany. There's a couple important deadlines coming up, so please consider making an abstract submission for that. We'd love to see you in August. I want to alert everybody about a couple of upcoming future Proteoform Thursday. On September 18th, we got Jenny Broadbelt from UT Austin. She'll be talking about a beacon of light development of UV photodissociation mass spectrometry for the characterization of intact proteins. October 16th, we're going to have a vendor showcase. And November 20th, we have Christian Bleiholder from Florida State University. While we're at it, I know that all of you saw the box on your registration form that had a place where you can suggest future speakers. I'm sure Paul and his team would love to have more input on that. And so if you have somebody that you'd love to hear from, please communicate that either in the registration form or send that information directly to Paul. So without further ado, let me again welcome Dr. Juan Antonio Vizcaino. Dr. Vizcaino is a distinguished scientist in the field of bioinformatics, currently serving as a team leader at the European Bioinformatics Institute. His primary research interests lie in the development of computational tools and resources for the analysis and integration of mass spectrometry-based proteomics data. He is well known for his contributions to the development of the PRIDE database and the establishment of the Proteome Exchange Consortium, which facilitates the exchange of proteomics data between different databases and researchers worldwide. He has been recognized as a leading expert in proteomics data standardization, and his work has been instrumental in the development of international guidelines for the reporting and sharing of proteomics data. He has been cited more than 30,000 times. Today he's going to give us a presentation entitled "The Implementation of Open Science Practices Can Enable a Faster Development of Top-Down Proteomics." Following the presentation, we hope he'll be willing to entertain some questions from the audience, and those questions can be submitted at any time throughout the presentation via the chat function. So without further ado, let's welcome Dr. Vizcaino.
Thank you very much, Michael, for the very nice introduction. It's really a pleasure to be here. I'm gonna be sharing the screen first. Let's see. Yeah, can you see the right screen?
Yes, sir.
Yeah. Yep.
Yes.
Okay, perfect. 1 second, and then I'm all set up. Yeah. Okay, good. So yeah, again, thank you very much for inviting me for Proteoform Thursdays. So it's really a pleasure to be here. So of course, at the end, I will be happy to discuss, or to even in the future. I mean, if you want to email me or just to follow what the topics I'm gonna touch base here, is also perfectly fine. Okay, so the title of my presentation is "The Implementation of Open Science Practices Can Enable a Faster Development of Top-Down Proteomics." So basically, this is the overview of my talk. I'm gonna give an introduction to the PRIDE database as part of Proteome Exchange. Then I'm going to talk a little bit of some examples of data reuse in the top-down proteomics world, just to show what is possible these days. Then I'm gonna kind of talk about FAIR data enabling the Human Proteoform project. Basically, some ideas that I've had for some time on that context. And then I'm gonna finish with a couple of important topics as well, okay? So first, I'm gonna talk about the Pride database and Protein as part of Protein Exchange. So I'm based at the European Bioinformatics Institute, which is located in Kingston, Cambridge, UK. And basically, EBI holds, I think, the largest collection worldwide of data resources. And my team is responsible for the Pride database, which falls in the category of proteins, together with PDB, or Protein Data Bank in Europe, and UniProt, and others. So since I'm gonna talk about FAIR data, I thought that at least at the beginning I need to put a slide about what FAIR means. Fundable, accessible, interoperable, and reusable. I think that this kind of term is becoming more and more popular. And one of the reasons why we do databases and databases like Pride is to make the data fair. So talking about fundable and accessible, Pride was created like 20 years ago now, actually, by Leonard Martens here at the ABI as a data repository to store proteomics data sets. And basically, Pride stores all types of all data types related to a proteomics experiment, including the mass spectra, raw data, peak list, vector and protein expression data. And really all proteomics approaches are supported, but some better than others because of different reasons. So at the moment, as of today, Pride contains approximately 48,500 data sets, just for everyone to realize of the magnitude of the data Pride contains. And very importantly, we are not working in isolation. We are coordinated. 6 resources worldwide, Protein Exchange, Beta Dallas, Massive, and Panorama in the US, but also iProx in China and JPOS in Japan, really implementing standard data submission and dissemination practices between the main proteomics repositories. And thanks to the reliability of Protein Exchange, really data sharing has generalized in the proteomics field. So what is included in a Pride or Protein Exchange data set? So it includes the original experimental MS data, so the raw data, the primary data, also peak list, identification results inferred from the original primary data, and quantification information. These are the so-called process results. Then experimental and technical metadata, and optionally any other type of relevant information like search database, or spectral libraries, or other data types. This chart shows the increase in amount of data submitted to Pride per year. So this is not cumulative. This is basically year after year. So we started Protein Exchange in 2012. At that point, we received 172 submission of data sets, submitted data sets in that year. In 2024, we received almost 7,300. So this is an average of 608 submitted data sets per month in 2024. And Pride really is the world-leading resource, storing approximately 80% of all data sets. And of course, this is possible because, we are the same amount of people, and managing all this is basically due to huge improvements in the infrastructure, focus on scalability, reliability, and automation. Of those data sets I've been checking today, and really top-down proteomics data sets are in the low hundreds. So I think there are around 200 data sets that are public, but there are kind of similar amount of data that is still private, because of course, data sets are private during the manuscript review process. Okay, so I wanted to give some examples of public data reuse that are happening in the bottom-up world, just to really show what could happen with top-down proteomics in the future. And this is the R of FAIR, so the concept of reusable data. So there are very different ways to introduce this, but lately, because AI is so trendy, I'm kind of introducing this as a way to make proteomics data AI ready, or to be used in AI related applications. So this is a figure that I kind of took from a paper from Matthias Mann from 3 or 4 years ago now. And basically, it summarizes all the stages of the proteomics kind of sample, and then analytical, and then computational analysis workflow. And shows the kind of, this is completely out of date now because there are many other tools now, but this shows some tools that have been developed using AI to improve these stages. So kind of the the first role of Pride and Protein Exchange in AI-related efforts is to provide reliable, fair data for the community, so enabling AI. And that means, to me, to provide a reliable infrastructure to submit and download data. And this means really the MS raw data, so the raw data, the primary data can be used straight away. This is more limited for other proteomics data types, but I will explain how we do it. And then in this context, I just wanted to highlight that there are really multiple AI approaches that are based on the reuse of just the raw data or the MS data coming from public data sets. And these kind of the approaches that have become more famous are the ones that are related to the prediction of MS2 spectra, so the prediction of peptide fragmentation. And then there are other approaches related to peptide and protein identification, like the use of the novel approaches or rescoring approaches or similar things. And again, these approaches, the development of these approaches have really been enabled by the availability of a large number of public data sets. So there are multiple publications. These are a little bit kind of, most of them are a little bit outdated, but I should update a little bit this slide to show kind of the latest publications, for instance, in the novel sequencing tools. So the second mission that I think we have, apart from provide reliable fair data and direct access to MS-RAW spectra, is to generate AI-ready data. So that means collections of reference data sets that are consistently reanalyzed, because we all know that there are too many ways to analyzing proteomics data sets. They need to be well annotated data sets, so the correspondence between the samples and the raw files. And there are many examples of this. Today, I have limited time, so I only touch on PTMs, protein expression values, but there are, of course, other options. And that's why also this acronym of FAIR is being kind of repurposed now to mean also fully AI ready. So I think that for this audience, I don't need to explain this, but I will just mention that apart from the actual raw data, for other data types, to provide AI-ready data for other proteomics data types, the data sets need to be reanalyzed. And they need to be reanalyzed in a comparable manner, because there are too many different approaches and tools to analyze the data, as we all know, and at least the analysis needs to be made comparable. These are the steps required that anyone will need to do in order to be able to do this. There needs to be a dataset selection step first, based on biological and technical criteria. The dataset manual curation, this is really required, especially to match the samples to the MS-RAW data. Then we need to have data reanalysis pipelines, including QC and also post-processing. And then what we also do, but of course this is kind of optional, is data integration of this generated data in other data resources. So these activities, of course, require often also research about the methodology, because we all know that proteomics approaches are very dynamic. So we have been doing this for a number of years. Again, I'm just going to show kind of what is possible to do today, what we are doing also in collaboration with others. So at the qualitative level, we are doing this for peptide sequences and PTMs. So a few years ago, we started the PTM Exchange project. The main objective of the project is to make PTM data fair. That means to have high-quality collections of PTM data and integrate this data in UniProt, linking to the original mass spectrometry order. So in one sentence, making PTM data fair. So the way we do this, we take data sets that are available in the public domain. Most of them are for Pride, and we reanalyze them, in this case, using the TPP pipeline from our colleagues in Seattle, Eric Deutsch, because this is a project that was started in collaboration with them. And then we have collections of PTMs that have been reanalyzed in the same way and with the same assumptions. So it's really PTM AI-ready data that gets integrated into UniProt. We have done what we call a lot of builds now. We started with kind of organisms that kind of provided small builds like rice or plasmodium, especially in phospho. We started with phospho, but lately we have moved to mouse and human phospho, and also to other modifications other than phospho. So we have done human sumolation, human ubiquitination, human acetylation, and we are also doing at present human methylation. So again, it's the same process. One has to go to Pride Protein Exchange to select data sets that are suitable for this. Then we use the same data analysis pipelines. The data set are curated. The kind of statistical post-processing is done in the same way. And this is the way we achieve this kind of consistently reanalyzed data sets. Then the data gets into data resources, so it gets into Pride, it gets into PetAtlas, and it also gets into UniProt, which was our main objective from the beginning, to really enrich the information related to PTMs in UniProt there. So this is just one example for one protein coming from rice. And one can go to UniProt and see there's kind of annotations for phosphorylation there, the origin of the sources PTM exchange, and then there is a link there where you can see the original spectrum, for instance, in Pride. So again, this is something that we have been doing and we continue to do because, as you can imagine, there are countless numbers of organisms and many PTMs. So this is really an ongoing project, and basically also many people are getting involved on this. The second way to provide AI-ready data, to provide a consistent and updated view of the proteome, is doing at the quantitative level. So also we have been doing that, and I'm not going to get too much into the details, but really what we wanted to do also is to integrate gene and protein expression information in the same interface. There is a EBR resource called Expression Atlas that enables now the visualization of both data types together. And again, we follow the same thing. We select a group of data sets that are kind of related biologically, or it's mostly biologically related, and then we analyze them using the same tool and the same assumptions, and at the end we integrate the data, in this case in Expression Atlas. So we have done many of these different studies in the last few years. I just want to highlight, for instance, that we have kind of perform protein expression studies in preclinical species, because there was a lot of interest from some pharma companies having this information coming from the main model organisms like mice, rat, and pig. And for this we reanalyzed like 37 public data sets in total from the 3 species. And also another project, so projects that we have done also that are similar, are protein expression in cell lines on tumor tissue. So we have integrated different experiments, including cell lines and the corresponding tumor type. Or we also recently, or more recently, last year, we did a tailored study for colorectal cancer for biomarker discovery and validation. So again, this is all possible because there are a lot of data sets in the public domain that can be reused for this purpose. Again, and this is kind of how the data looks in Expression Atlas. So in this case, you can see the proteomics data sets there, and this is just how the expression matrix looks like. So this is really what we are doing more and more. You can see Pride here in the center, and then using the same approach, we are kind of generating consistent reanalyzed data that gets integrated into other resources. I have shown the sample of UniProt for especially PTM data, but also Expression Atlas for quantitative data sets. But we are doing similar things in other contexts for proteomics data, for instance, metaproteomics and metagenomics, and also for protein structural data. I will explain something of this at the end of my talk. Okay, so after this introduction, where I have shown what is possible to do, what Pride is, protein exchange, and some examples that there are many others. I just have a few minutes to give a few examples of what is possible to do today. I'm gonna get into the kind of the main section of the talk today, and is how FAIR data enables or could enable or enables the human proteoform project. So for those of you that don't know, the Human Proteome Project was started by HUPO, the Human Proteome Organization. I think it was in 2010 in the conference in Sydney, and since then it has been going, trying to find at least one proteoform or the canonical protein for each gene in the human. Basically, the way it works is that, or one of the ways it works is that people that are working in that project, they go to Pride or other protein exchange resources, and they choose public proteomics datasets there because of different characteristics. It could be biological reasons, it could be technical reasons, and then they perform a dataset selection, and then they do the same process that I have tried to explain before. They do data reanalysis by QC using open software, or free-to-use software using the same reference system, the same search database. In this case, UniProtDB used to be before Nextprot. Then the results, they go through community guidelines for interpretation. It means that they basically summarize what is a good protein identification in the context of the Human Proteome Project. And then every year they provide like a new build of what is considered kind of the community accepted list of detected human proteins. So this is just the paper that was published, I think, a few months ago about the 2024 status of the human proteome. And at that point, there is evidence for 18,397 human proteins, 93% more or less. The HPP was initially organized per chromosome, and I think it worked very well in some aspects. Of course, other aspects it didn't work so well, but at least it was a very good exercise. It is a very good exercise to come up with a community accepted list of detected human proteins. So actually, I think I took this logo from one of your websites. I don't know if it's updated or not, but I took it. So the Human Proteoform project, at least one of the branches of the project, could work in the same way. So one would go to Pride Protein Exchange and find public proteomics top-down data sets there. There could be a data set selection. There will be a data reanalysis plus QC using open software or using free-to-use software, using the same reference system, community, and then the results, they will need to be filtered based on community guidelines. And I will go through it again in the next slide because I cannot stress enough how important is to have these guidelines could be. Then the actual list of proteoforms, they could go into resources like the Human Proteoform Atlas that the guys from Niels lab have developed very nicely. And then also adding sample metadata information, we could also have protoform AI-ready data. So basically the same concept as before. This same kind of concept could also be used for newly generated data. So of course, so far there are not so many data sets in the public domain, and the quality is quite heterogeneous, but the same could apply to newly generated data. Again, it would be reanalyzed using the same software, and the data would be interpreted using the same community guidelines. So I cannot really stress this enough. I think this is something that I have mentioned the first time several years ago, is that community guidelines are essential to advance the field, the top-down proteomics field. What is a good protoform identification? So this needs to be sorted out by the top-down community. I mean, because non-experts in the field need to know how to be able to reuse the data in a meaningful manner. Again, these are the Human Proteoform Project guidelines. This is the version 3.0. So I think this is the last version, was published like five or 6 years ago. That means that, of course, before there was a version 1.0 and a version 2.0. So no one expects that the first version will be perfect. So they can evolve. And then also the guidelines can be implemented using a checklist. That is what is also provided in the paper. Of course, there are some things that are very important as well. In the context of top-down proteomics, ambiguity is also a very important factor as well. That should also be part of the guidelines. So of course, this paper was, I think, led by Lloyd several years ago. And this, there are different categories here depending on the ambiguity of the identification. But let me stress this again. Community guidelines are essential. What is a good protoform identification? So the guidelines should include approaches and statistical thresholds. So for instance, maybe visual validation is needed, at least in some cases. And without this, protoform identifications cannot really be propagated across data resources, for instance. The guidelines will need to be dynamic anyway, so they need to be versioned, but they should be completely open and developed in an open manner with open participation. And also, ideally, it should be possible to validate automatically some aspect of the guidelines using software. So if these guidelines exist and it is clear for everybody what is a good protoform identification is, at least as of June 2025, then reliable protoform identifications, detections can be propagated across data resources. And this is one of the things that we have been aiming for years, to have this proteoform atlas companion system, the terminology I think that was invented by Harmut, basically where interoperability is possible between different types of data resources. In the center, we would have the proteoform-centric data resources, like the human proteoform atlas, but also the proteoform ID registry and other resources from the consortium of top-down proteomics, for instance. On another side, we will have, for instance, Sprite to store the mass spectrometry data, experimental data, the primary data that, it could be reanalyzed in different ways to feed the information that goes into the proteoform-centric data resources. And also in another branch of this system, it would be like the protein-centric data resources that are accessed by biologists, no? So UniProt or Intact or Reacton or PDBE, for instance, because it's also essential that, scientists that are not, or biologists that are not expert in proteomics, in top-down proteomics, can really access this information. So for this, of course, interoperability is needed. And one key component that is needed, and again, we have been discussing this for quite some time, is that we need agreed standard identifiers for proteoforms to enable interoperability between resources and tools. At least the minimum that we need is standard identifiers for proteoforms. But again, if we know what a good proteoform identification is, then really we can fit information across different types of resources. Then again, if these guidelines exist and we have clear what a good proteoform identification is, then we can also generate AI-ready proteoform-centric data. And also we can integrate it with bottom-up proteomics data, like what I have been mentioned before, for instance, in the context of the PTN Exchange project. But because of having AI-ready data, it would be possible to do things like are happening in the bottom-up proteomics, or like rescoring proteoform spectrum matches. So this is just a paper that was accepted a couple of months ago. I haven't read it in detail, but I saw that what they do is that they are trying to rescore proteoform spectrum matches using deep learning approaches. But of course, for this you need to have a very good benchmarking data. Then, if we have also clear what a good proteoform identification is, we could provide other types of performing data analysis, for instance, by generating a spectral library. So also I saw a recent paper in analytical chemistry. I think this is for last month. The top-leap system, where again is the same idea that has been going on in the bottom-up world for many years. You have a lot of data in the public domain, metadata and raw mass spectra, and then you use the same software and the same assumptions to analyze the data, and then to build at the end a representative spectral library. So again, this would be enabled by that as well. But I mean, these are just some ideas. There are really many other possible options. And I've seen that there are some people reusing more and more also top-down datasets, but of course, the number of reuses is really still very small in proportion. So I have another couple of topics to cover in my presentation before finishing. Is what I call other important topics. The first one is the importance of having open or freely available software for analysis and QC. So again, we come back to my previous slide about how the Human Proteoform project could work considering public proteomics data sets. Then it is clear that we need to have open or at least freely available software. Now, of course, the requirements of that software is that it needs to be free to use, it needs to be reliable, so of course it needs to be good quality. It doesn't really matter much whether it is open source or not. I mean, open source maybe is a bit better if people want to check the code and everything, but what it needs to really be is to be free to use. Because that means that, some people can try to repeat the same analysis. The software needs to be versioned and well documented, and ideally it should be possible to use it in HPC environments, either in-house or in a cloud somewhere. So in-house, it means in a kind of farm, like for instance, the one that we have here at the ABI. And if we have the same kind of input data, the same data set, we have an open software that is also versioned, then those are kind of the pieces that are needed to have an analysis that is reproducible. So in that context, we did a small contribution a couple of years ago, also with the collaboration of John and also Dave Tapp. This work was led by Matthias Walser, that at that time he was in my team. And then basically we developed open software, what we call top-down app, basically reusing a lot of kind of open source software that is used for top-down analysis, especially topic suite here. And this is what we try to achieve. So we try to achieve to have like one platform that is open source that everyone can use in order to reanalyze top-down proteomics data sets. Of course, I know that the software kind of is very dynamic and evolves all the time. And this is kind of of course, one of the challenges of having this kind of platforms that for data analysis. But this is the idea. The idea is to have software that is made available to everyone in the community. And with that software, one can reanalyze data sets in the public domain or data sets in-house, making the analysis reproducible. Okay, and the second topic that I wanted to cover is that how top-down proteomics data could look in Pride in the future. So, and for that, I need to explain a project that we are currently doing in collaboration with the cross-linking community. So I have shown this slide in previously where Pride is in the center, and then we are trying to feed data from Pride into other data resources. In this case, we are trying to feed protein structural data into the protein data bank in Europe, also in the protein data bank in the US, in PDB IHB, and integrated hybrid models. And then we started a project called 3D Proteomics, where we wanted to provide improved support for cross-linking datasets in Pride with visualization capabilities, and also improving the standard data formats for cross-linking. And really the objective was also to link datasets between Pride and PDB at submission time as well. So really the broader aim of the project was to bridge the gap between proteomics and structural biology data, including guidelines and data formats. And we have now a version 1 of the Pride cross-linking resource. And this is in this URL. You can go to Pride and go to a subset of Pride that is devoted to high-quality cross-linking data sets. What we provide there is a bespoke view for cross-linking data. And as a key point, we do it in collaboration with a group who is expert in the field. So in this case, we are doing it in collaboration with a group from Julie Rapsilver in the University of Edinburgh here in the UK, but also now in Berlin. And basically what we do is to create a section of Pride for cross-linking data, which also provides a visualization tool that is implemented next to the data in the Pride infrastructure, cloud-based environments. In this case, the tool is SciView. Again, it's a tool developed by Julie Rapsilver's team, and this is basically just a screenshot of how the cross-linking data can be visualized there. An important concept there also is that in order to be able to read cross-linking data, we are supporting a standard data format. So in this case, MCAD-NTML 1.2 and 1.3. I'm not going to talk about data standards in this talk today, but it would also be great if the top-down proteomics field kind of supports better MCAD-NTML or NCTA, because that would also enable a lot of functionality in the future in that context. So this is really a model for us to, something I have forgotten to mention is that Pride cross-linking also provides links to UniProt, PDBE, and AlphaFold. And this is really a model for us in the future for other proteomics approaches. So maybe this could be a model that we could use in the future for top-down proteomics. So what we would have is a subset of Pride devoted to high-quality top-down proteomics datasets. The dataset ideally should contain the results in the same format, ideally a standard format. It could be MCAD-NTML, for instance. Then we should have a visualization tool that is tailored for top-down proteomics data, maintained in collaboration with the community or with the research groups in the top-down proteomics field, but deployed in the EBI infrastructure. We can, as you can imagine, we cannot develop a tool for every kind of type of proteomics data, but we can reuse the tools that are developed by others, and we can deploy that in our cloud infrastructure next to the data. Of course, that would be like a collaboration between us and the group developing the tool, because of course the tool is not just deployed once and doesn't change anymore. It kind of involves a dynamic development. Then ideally the dataset should be well annotated, for instance, using the SDRF proteomics format that we are adopting. And this is really a model that works very well for the future for many subdomains. So because this creates really a synergy between Pride and the domain specialist groups. So for me, in my opinion, really everybody wins. And also what we should also do with this infrastructure is to link the information to other sources in a proper way. Okay, so conclusion, just to finish my presentation, I hope I could explain to you that open science practices are really the way to go. In proteomics, a part of protein exchange, Pride represents the current state of the art. Top-down proteomics, in my view, could really benefit enormously from a wider implementation of open data practices. More datasets in the public domain, which could be reanalyzed in the future, and they could become a key component of a human proteoform project, together with newly generated datasets. Community guidelines are essential. Hopefully you got that message. What are good protoform identifications? This is key for being able to propagate information, especially for non-experts in proteomics, so for general biologists in databases like UniProt or PDB. This would also enable the generation of community-agreed AI-ready data and spectral libraries and many other applications. And I have also mentioned the importance of free-to-use software for enabling reproducible analysis. And then I've also talked about maybe some idea for the future of having a future section for top-down proteomics data in Pride. And I would really like to thank everyone in the Pride team, and also to all our funders and collaborators. Yeah, thank you very much. Happy to take any questions.
That was a really fantastic seminar. I have a whole page of questions like written down. Yes, Neil, let's clap. Let's clap. Let's cheer quietly in our office.
Virtual and real.
Yeah, exactly. So I believe that the chat function is available. We'd love to see questions from the audience come in there. What we can do is, if you put a question in there, we can unmute your mic, and you can ask your question live, or I can just read it. Maybe to kick things off, while we're accruing questions, I can ask you a couple. Several times you used the term AI ready, but you didn't really get into that. Can you go down one level deeper, so that, let's say, I don't actually have any AI skills, but let's say that I did, and I wanted to take advantage of some AI ready information from Pride. Like, what does that look like right now, and what do you anticipate that it's going to look like in the next few years?
So at the moment, it is not, so let me first talk about what I think AI ready data is. So AI ready data means data that is kind of generated in the same manner, again, because the data needs to be comparable. So at least for processed data, at least in my opinion, it means that at present it needs to be processed in the same way, at least with the same assumptions and the same protein sequence database and similar things. And then it needs to be ideally well annotated. So one needs to know, this MS-RAW file corresponds to this particular biological sample and these kind of things. So, and a last part of this, it needs to be in the right data format. So especially if we want to use, a lot of kind of this data becomes very big in the future. So the last part of the data format is kind of a work that is starting now. There are other databases that are starting to use the so-called embeddings, and I think we are just starting now. So in the PSI, there is a working group that has been just recently created that is going to work on this. Okay. So, and in the context of then the AI readiness, it means again data that has been preprocessed in the same way. And yes, I have to say that the information at the moment is not kind of very well centralized, because the problem is that there are a lot of people contributing to this, and there are different aspects, PTNs, quantitative information, all these kind of things. So this is something that we definitely have to improve, but this is what and this is what it would mean. I don't know if I was clear in my response.
Yeah, no,
Yeah.
absolutely. It really helps a lot. And it's something that I've been thinking about a lot because um, the the impact of deep learning in mass spec and proteomics has already been clearly demonstrated, and I think it's quite reasonable to assume that the more people engage with this, the more talented people engage with this topic, sky's the limit with what can be done both in bottom up and top down, and the integration of the two as far as like deep learning and AI goes. And so I commend you for stepping forward and thinking about how to make information available so that we don't have to download the entire Pride database every time we want to do something AI, that the data is already going to be in a format sort of amenable for use in various sort of downstream applications. um Looks like Neil is spamming the chat. Neil, do you want to propose a few questions out loud as long as you're up and on screen?
Well, sure. Yeah, and thanks, Juan, for that. And the reminder of just what's so important and fundamental. And I circle back to what other areas. So you've got your fingers on the pulse of the field, and um, even if you just talk about compositional proteomics, not spatial or single cell, for just good old-fashioned composition of the proteome, say even this the human, what other areas have outcompeted because there are finite resources, and we do aspire to the Proteoform project, which would bring the kind of resources necessary to implement. You're so clearly the roadmap you laid out is needed. And then I circle back to, well, what other areas have allowed competed? What's the composition of pride presently? Like, because you could say you might say 1% is top down in your pride right now, or you could say 10 or growing. I don't know. But it's really the resources to implement your vision there. And why haven't we got there? How can we get there in the future?
Okay, so I think I mentioned before that I think there are like between 400 and 500 data sets in Pride that are top down, or at least that are annotated as top down or somehow top down. Then one needs to check really if the annotation is right. So I would say that is approximately now like 1% of data sets.
1%. Yeah. Okay.
I think that's not a bad estimation of that. So I think, but maybe I don't have really the hardcore numbers to back this, that the top-down field kind of is more reluctant to share data in general than other type of proteomics approaches. I think top-down proteomics accounts for more than 1% of what the data is produced today, but maybe you have a better idea about around that. And your question about the resources that are needed, I think that as a starting point, I mean, again, you don't need to see this as a, you know, yes, we are going to start this tomorrow and then everything will work perfectly from day one. I think that it needs to be implemented in a gradual manner. So to have like a proof of concept of how this could work, selecting, I don't know, you must know the five or top 10 kind of best data sets in the top-down proteomics world, and chose those 10 data sets and come up with, version 0.2 or 0.1 of, community guidelines. That to me is something that it is, I mean, it's work, but it could be done by a committed working group that kind of that meets one every couple of weeks or every week. I mean, this is the way that many of the PSI standard kind of efforts have happened. This is the way we did, for instance, Proforma 2.0. We kind of met every couple of weeks for 2 years to do it. So while I agree that for implementing the whole vision, it is quite a number of resources that are needed. I think that to have this kind of proof of concept and coming with a first kind of version of what the community guidelines could be, I think that they would need, of course, some commitment would be needed, but I think that it's not it would be perfectly feasible because this is the way we have done other kind of similar things.
Great.
That's what I think. It
That's
requires another thing that is very important. It requires a champion. It requires a person or a person or two that are really the ones that are going to be pushing the others. So that's also very important to have. I forgot to mention that. You need at least one person, that is gonna take the kind of the responsibility in pushing things. It's not because the others they don't want to do it. It's because everyone's very busy and there needs to be one person always or two that
Beating the drum, yeah.
that are the champions for this.
that was a really outstanding answer, I think. Juan. Couple more questions like that I'm very eager to hear answers to. One is by Paul. I was part of this workshop last week, and there's a good handful of software packages now available for top-down, even though the variety of answers we get still continues to be non-overlapping. What are your thoughts about choosing top-down software packages to sort of represent results in Pride top-down and Proteoforms and stuff like? Is that part of a community effort? What are your thoughts there?
So to me again, it should. I mean, I don't think it's more about selecting software A or software B. It's more selecting about the approaches and being kind of do the same assumptions. So at the moment, I know that there are kind of, again, I'm not but I know that there are kind of two main schools. Kind of someone goes to from the kind of the concrete protoforms, and some others that they try to get first one particular protoform and then maybe use that as the seed to come up with the others. So I mean, there are different ways. Maybe for this pilot project, since it's starting and you need to choose something to start with, maybe to make it less complex, one would need to maybe start with just choosing kind of one of the main softwares and the main approaches. I think maybe, I mean, again, or if people think that they have the bandwidth to do it with two, then also, I mean, that's not a bad idea. But 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 the main thing is that people shouldn't feel, that you are choosing my software over your software because it's better or it is worse. It's because, we need to use something. And
Yeah.
it needs to be clear, that we have chosen, or maybe the working group, if there is a working group, and that's a very important point. In the working group, there are representatives from two different softwares. They are going to be the best that they know their own software. So then it would make perfect sense to use those particular softwares there. The important thing would be to document again the assumptions and the way to do it.
Yeah, I think there's a theme here between the answer you gave me and the one you gave to Neil, which is that let's not wait until we do everything perfect. Let's do something. Let's get started and let's have some discussions and then build on that. We'll sort of build some momentum. So I think that's a fantastic recommendation. Claire Boos has got a question. Claire, let's see. I'm going to unmute you. And let's see.
You can't read my question yourself.
No, go ahead, Claire. I think you're the perfect person to ask it.
Yeah, I guess it kind of bounces off what the previous discussion is about, which is like, can you incorporate proteoform classes into how you're going about binning top-down data so we don't just throw out like IDs that don't necessarily, localize and identify post-translational modifications, but it's still useful data in the sense that you have potentially a protein ID with a mass shift. So I'm just wondering, would there be a way to take top-down data and just bin kind of by class so that we just accumulate all that data, but maybe prioritizing it differently?
Yeah, that definitely, I mean, I see this as a possible approach. If this is what, the state of the art is, it's better to start with something that, that people feel more confident and use it as a first stage and then go for, other type of more ambitious objectives for the future. So that's a very sensible approach.
Great. So So we have one
For
or two
instance, to
minutes.
put an example, for instance, no, yeah, just I'm thinking that, for instance, at the beginning, phosphorylation data, for just to put in bottom-up approaches, I mean, there were very few people using the, the software to do the ambiguity in the modification position, the phosphorylation position. And now, several years later, it's much more common to use this kind of post-processing software downstream. So this is just an example. I think that maybe is a little bit analogous to what you were trying to express, Claire. So that would be, I think that would be a sensible approach.
Yeah, your reanalysis of bottom-up phospho data was eye-opening for the field. The 40% or some number around there of phosphosites that were ambiguous. I mean, really, I think put a big shining light on the ambiguity question.
Yeah.
And Claire, could I get you to introduce yourself just a bit? Where are you working out of? And yeah, if you don't mind.
Oh yeah. I work with Dr. Shortreed. I'm in Lloyd Smith's group.
I see. Okay, great to know you.
Yeah,
Nice to meet you.
Claire is our top proteoform identifier. So watch her star rising. Anyway, so that was I think the questions have slowed down in the chat, and I think we'll wrap up here. But I want to thank you.
One more thing,
One
Mike.
more, Neil.
Yeah, just that. So under HubMap, and so resources tying to community development, Liliana's group in PNNL and our group, we combined the software and created a combo, synced our false discovery rates around that, and then reanalyzed some. And that sort of was underneath the HubMap project of NIH back a year or two ago. I mean, it's a baby step, but it is a step that was taken. Sorry, Mike, go ahead. Why don't you take us?
No, that was great. That was important. I'm glad you said that, Neil. Yeah, so let's wrap up. This is a fantastic day. Thanks, Juan. And I hope to see everyone that's on this call at the next Proteoform Thursday.
Okay, thank you very much. As I said, if you need my help for any of this, I'm always happy to help.
Fantastic.
Okay.
Thanks,
Have a
everyone.
great summer. Yeah.
Good. Thank you very much, everyone.